bueno, evidentemente por razones de horario, pues tuvo que cambiar de nuestra hora habitual. Pero lo importante es que vamos a escuchar un excelente seminario y bueno, me voy a permitir presentar formalmente al doctor Iñaki Ruiz. Él es biólogo de la Universidad de Barcelona. También obtuvo su doctorado en biología en el Departamento de Genética, también por la Universidad de Barcelona. Y en el 2003 obtuvo una beca postdoctoral de la EMBO para hacer su postdoctorado en la Universidad de Luis en Canadá, en el Departamento de Bioquímica y Biología Molecular. En el 2006 consiguió otra beca postdoctoral de la Fundación CSIC-Universitat para extender su estancia postdoctoral en el mismo departamento, en la misma universidad, por otros 2 años. Y en el 2007 se reincorpora a la Universidad de Barcelona ya como un investigador y CREA. En esa posición estuvo del 2007 al 2011 y en el 2011 ya se convierte en un profesor investigador en la Universidad de Barcelona, también con en reconocimiento y CREA. Y evidentemente el interés de la investigación del doctor Iñaki Ruiz pues tiene que ver con la multicelular de los organismos animales. Y en ese sentido, pues realmente es sorprendente las contribuciones que ha realizado y publicado los resultados de su investigación de su grupo en revistas pues muy de alto reconocimiento como Cell, Nature, Science, etcétera, etcétera. Ha formado múltiples estudiantes de doctorado y postdoctorados. Y pues realmente le agradezco mucho a Iñaki que haya aceptado la invitación y que esté aquí para darnos el día de hoy el seminario que tituló The Origin of Animals, a Black Swan Event. Adelante, Iñaki, muchas gracias.
Sí, muchísimas gracias, Gustavo, por la introducción y bueno, bienvenidos a todos. Voy a compartir pantalla primero de todo. Vale, buenas tardes a todos. Y en teoría ahora está en formato grande. Y bueno, voy a pasar a inglés, pero si preferís en castellano, lo dejo en castellano. Simplemente lo tenía pensado hacer así por si acaso, como me dijiste que estaba online y todo. So what I'm going to do is I'm going to explain what we have been doing in our research, in our lab, in order to understand the origin of animals. The question that we have, it's very basic. Well, it's basic to us, not so basic to answer. Basically, we are amazed by the huge diversity of animal body plans, their behavior, their embryology. But we want to understand more than those things. We want to understand how all these appear. So basically, we want to understand how, at some point, unicellular organisms, apparently as simple as those ones, could eventually give rise to the huge diversity of animal body plans that we see today. And in order to understand this question, there are 3 important points that we need to consider. The first one is how was the unicellular ancestor? The second one is how were the first animals? And the third one, which you can only address if you have first resolved the other 2, is how the unicellular ancestor became the first animal. The best approach for that is comparative genomics. So actually, several labs were already answering how were the first animals, but there were almost no people, no researchers working on how was the unicellular ancestor. So we wanted to elucidate that. And the best approach is comparative genomics. We basically compare the genomes of animals with the genomes of their closest unicellular relative. So the unicellular relative that are genetically closest to animals. And by comparing the genomes, you can infer how was the genomes of the ancestor, if it was complex or not. So basically, the idea is if the genomes of animals have gene A and the genomes of unicellular relative also have gene A was present in the ancestor. If gene B is only present in the genome of animals and not present in the genomes of unicellular ancestor, the unicellular relative, then it was not present in the unicellular ancestor and was a genetic innovation in the branch leading to animals. And the thing is, you may say, what, but there is no already a theory that explains how animals evolve. And yes, there is a theory. There is a wonderfully very nice and amazing theory, which I'm going to explain now. And after I explain it, I will try to destroy it. Okay, so let's start with this theory, which is the classical theory. This classical theory is based on three assumptions. Three. The first one is that the sponges are the sister group of animals. So that this group of animals that are morphologically simple are the sister group to the rest of animals. And actually, this makes some people wrongly, because you cannot infer that, like this, that if sponges were the earliest branching animal, then the first animals should be like sponges, which is something that you cannot do because there have been 6 million years of evolution, and you don't know if extant sponges were like the ancestral sponges. That's the first assumption. Second one is that choanoflagellates are the closest unicellular relative of animals, which again made some people believe wrongly that means that the unicellular ancestor was like the extant choanoflagellates. Again, you shouldn't be doing that, but a lot of people did that. And the third assumption is that there is an homology between choanoflagellates and one specific cell type of sponges. This homology is the basis of the choanoblastia theory. So basically, you had an ancestor, unicellular ancestor that looked like extant choanoflagellates. It gave rise to the first animals that look like extant sponges, and from sponges, the huge diversity of animal body plans. So this choanoblastia theory, actually what proposed is that you had choanoflagellates, and then you have choanoflagellate colonies. And these choanoflagellate colonies will become more complex and give rise to what it was called the choanoblastia. And then more advanced choanoblastia with some kind of cell differentiation, and then the sponge, and then the rest of animal diversification. This theory, if it's true, it has two important implications. Actually, three, I changed that. Three important implications. There was an important genetic innovation just before the origin of animals. The origin of spatial cell differentiation was just at the transition to animals, and obligate multicellularity was just at the origin of animals. This is a very nice story, but maybe it's too nice to be true. Now, let's revise these three assumptions. The first one is that the sponges are the first animals. The thing is that there has been recently a lot of phylogenetic analysis that shows that ctenophores, and not sponges, may be the earliest branching animal. And ctenophores are morphologically much more complex than sponges. And importantly, they do not share that cell type that resembles choanoflagellates, this potential homology. The second assumption is that choanoflagellates are the closest unicellular relative of animals. And this is still true, but we know also that there are other several unicellular relative of animals, closely related to animals, that are completely different from choanoflagellates. So any of those, or the ancestor could be like any of those. And the third point, that choanoflagellates as a sponge share a specific cell type that was considered to be homologous, while recent data, both morphological and molecular, has put this into question. Okay, so we go back to these points. Unicellular ancestor, how was the first unicellular ancestor, how were the first animals, and how the unicellular ancestor became the first animals. Then we want to go to the first step, how was the unicellular ancestor, and we need to compare animals, the genomes of animals, with the genomes of their closest unicellular relative. One of the problems we had several years ago is who are the closest unicellular relative of animals. That was not known 20 years ago. So the first step was to resolve that. And we did that with several phylogenomic analysis, by doing phylogenomic analysis of all the eukaryotes. And we showed that there are 4 unicellular lineages that are closely related to animals: choanoflagellates, philasterians, ichthyosporeans, and Corallopithecines. Because I know that the names are difficult, I have also put 1, 2, 3, and 4. 4 lineages. I'm going to revise those lineages before we go to the real data. So the first one are conoflagellates. We know here that there are around 100, 150 known species of conoflagellates, and there are unicellular flagellates, free living in freshwater and marine environments, and they can form, some of them, they can form colonies by clonal division. So you have one cell, and then you have two cells, and then you have four cells, and so on and so on, like this timeline video. This is the way that they develop into colonial colonies. The second one is called the Philasteria, with only five known species. Those are completely different from conoflagellates, freshwater, marine as well. Most of them are free living. Some of them it's not sure if they are free living, and they all are amoeboid with phyllopodia. The best known is Capsospora osataki, which was isolated from inside the hemolymph of the freshwater snail Bionphalaria glabrata. And then we discovered that it has three different life stages. The first one is the normal phylopodial stage, in which it uses the phyllopodia as branches to attach to the substrate. This is in normal culture conditions. When there is not enough media, then the phyllopodia are retracted, and then you have a cystic stage. And then you have an aggregative stage. So at some point, the cells will suddenly come together and form an aggregative structure. Inside the aggregative structure, there is some kind also of extracellular matrix. So as you can see here, picture D, there is some kind of extracellular matrix in between the cells. We go to number 3, Ichthyosporea. The Ichthyosporeans all go through a specific, this is for example Spiroforma artica, which was isolated from inside Gammarus setosus, and they all go through this specific development, in which they start with one cell and one big white cell wall and one nucleus, and then the nuclei will start to divide without dividing the cell, and it will be a cell with several nuclei, up to 128, for example. And at some point, they will cellularize each nucleus. They will create the cell membrane and the cell wall, and then the external cell wall will break, and all the cells independently will go to the new, they will go to the media and they will start again the whole process. So this is, I'm going to show a time-lapse video that shows what I just explained. So if you focus on the big one with the black arrow, you will see what's going on here. This is already mature, so it has already several nuclei. And you will see the nuclei are a little bit whitish, and they divide, and at some point they will go to the periphery. Once they are in the periphery, like now, they will create the cellularize, they create the cell membrane, the cell wall. And now the external cell wall will break, and the cells will be released to the medium. And there are other flavors of this. So for example, we have another species called Chironimus fragantissima, in which it breaks, but they have not formed the cell wall. So they have only formed the cell membrane. So they are motile. They have this amoeba stage that they can use amoeba stage to go to a new place and start again. This is number 3. Then lineage number 4, actually, also in the Spiroforma artica, we found also that there is one stage of this multinucleate, one moment in which it creates a polarized layer of cells that resembles quite a lot an epithelium. So you will see it now. Yeah. So that was number 3, and then we go to number 4. And you will realize that this organism resembles a little bit what was shown before. So actually, there are only 2 known species, these clades. And those species can either go through a normal division, or they can go into a multinucleate, like the previous one. So if we go to this one, the question is, which one would you sequence to understand animal origin? So actually, yesterday I asked this question to the class, and they answered very well. So actually, you had several options. You could sequence the conoflagellates because they are the sister group to animals, so the closest one to animals, and they do colonies. Or you could say, well, I would prefer to sequence the phylasterians, because they aggregate, they have amoeba, they are very motile. Or some people will say, oh, I would prefer to sequence the Diosporians. So, because they have this multinucleate structure, it's kind of multicellular as well. It creates this kind of epithelium, which is something very specific to animals. And some people will say the corallopithecians. But then the real answer is why not all of them? And the reason for that is because two main reasons. The first one is because they are completely different, all of them. And they have completely different developmental modes. And none of them can be used as a proxy for the ancestor, because we have more than 600, 700 million years of evolution. And the second reason is because secondary loss is very important in eukaryotic evolution. So I'm going to show two examples of how important it was to have all those taxa. Two kind of gene families. So actually, we obtain now 16 genomes of unicellular relative of animals, 2 of them by the lab of Nicole King in UC Berkeley, the other 14 by our lab in Barcelona. This is some of the results I'm going to show you. The first one is the integrin addition system, which, as you may know, is one of the most important addition systems in metazoans. It actually binds the internal cytoskeleton of the cell with the external extracellular matrix. So very important for tissue addition. So integrins, integrin alpha, integrin beta, and all the other components of the integrin antisense were thought to be animal specific because they were not found on the genomes of guanoflagellates, and they were not found on the genomes of any other eukaryote. But then when we use our taxa, phylasterians, septiembre, and corallopithecians, which was not used on this first analysis, we show that the origin of integrins was much earlier, and integrins were secondary loss, the bull process, in guanoflagellates and in fungi. And something similar happened with several transcription factors. When we had the analysis only with guanoflagellates, we showed that, or it was shown that NF-kappa, ETS, SMAD, nuclear receptors, grain-head, TBOS, and RUNX, which are all very important developmental transcription factors, were shown to be specific to animals because they were only present on the genome of animals. They were not present in the genomes of guanoflagellates or plants or algae or fungi or any other eukaryote. So when we incorporated phylasterian genomes and the genomes of Septiembre and corallopithecians, then we showed that the origin of those genes was much earlier, and they were, again, secondary loss in guanoflagellates. Still, some gene families like ETS, SMAD, and nuclear receptors, they seem to be specific to animals, but some of them originated before, something that we could have not unraveled if we had only used guanoflagellates. We had to use the 4 different lineages. So this is the view before our data, so actually several years ago, but after the guanoflagellate genomes, in the genes that were thought to be specific to animals, several transcriptional regulation genes, several genes involved in cell signaling, that's KITS code, hippo pathway, protein tyrosine kinase, TGF-beta, nodes, WING, and several addition families, including integrins. And this is the view that we have now. There are several gene families that seems to be specific to animals under the current taxon sampling, but there are much less. So that means that the ancestor was quite complex. So when we analyze further this and we compare the difference between animals and fungi, and we did an extra analysis also obtaining the genomes of several unicellular relatives that are close related to fungi, what we observe is that most of the difference between animals and fungi occurred before. So from the last opistocon ancestor, from the last shared ancestor between animals and fungi and the unicellular relative common that are relative to both of them, we observed that early, very early, there was a gain of multicellularity genes in the branch leading to animals and a loss of metabolic genes. And then it was a loss of genes of multicellularity genes in the branch leading to fungi. So that shows the importance of the past for evolutionary transitions. Sometimes in evolutionary transitions, such as the origin of animals, we tend to focus specifically on the transition. but it's very important to understand all the past history, which situates, it provides the raw material for the emerging properties that will appear later on. So after these first years of genome data that we obtained from those organisms, we showed that the unicellular ancestor of animals was genetically quite complex. And of course, much more complex than previously known or thought. The question is, what are those metazoan genes doing on those unicellular taxa? How they were co-opted, because it's clearly that co-option was playing an important role. And I'm going to show two examples here. The first one is brachyury. We showed that the genome of Capsospora has the gene brachyury. And brachyury in animals is involved in gastrulation. So it has a very important role in this specific moment in animal development, in which the three germ layers are formed, and the mesoderm are formed, and the internal organs are formed, and so on and so on. We observed that there was one homologue in brachyury in Capsospora that was actually quite conserved. So if we check the amino acid motif of the Capsospora brachyury, which is the one with the black rectangle, and we compare with brachyury and other T-box genes, which is the family T-box, we observe that it will have the same amino acid motifs as the animal's brachyury. So we decided to check, because we didn't have genetic tools at that moment, we decided to check whether that homology was so conserved to rescue the function. So we decided to go into Xenopus, collaboration with José Luis Gómez Escarmeta in Sevilla. Xenopus is one of the organisms in which development and brachyury and gastrulation is better known. So this is an adult of Xenopus, and here you have an embryo Xenopus. This embryo is perfect, it's normal, gastrulation has worked, brachyury has worked, and the way we know that brachyury has worked is because we see these violet lines, that those violet lines are myodi, which is a gene involved in muscle. But the important thing is that gene, myodi, is a downstream of brachyury. So if brachyury is working, it activates myodi. And we can see myodi, brachyury is working, gastrulation is working, everything is working. But if we do a double mutant of brachyury, then nothing is working. There is no gastrulation, brachyury is not working, there is no myodi. We can see something green. This is not violet, it's green, and this is to mark the blastopore is completely different. So there is no myodi, it's not working, nothing is working. If we go to this double mutant of brachyury, Xenopus brachyury, and then we inject the endogenous Xenopus brachyury, we can recover the function, and we have a normal embryo. I know that the shape is a little bit different, but it's considered normal by embryologists. So brachyury is working, you can see the violet lines there, myodi is in everything. Everything is working. We just recovered. We had a double mutant, we injected the endogenous one, and it recovered. And the important point is that the same happened if we injected not the endogenous Xenopus brachyury, but if we injected the Capsospora brachyury. If we injected the Capsospora brachyury, we had the same phenotype as if we injected the Xenopus brachyury. And it was able to activate myodi, which, interestingly, myodi is not on the genome of Capsospora, but Capsospora brachyury, it has the amino acid motif, it has everything to activate myodi. So we have one of probably the first example of a gene from an amoeba, from a protist, from a unicellular organism that is able to rescue a function in a completely bilateral normal metazoa, multicellular metazoa. This is one example. Second example, actually, then we did CSIC of brachyury, and then we compared the downstream genes of Capsospora brachyury, CSIC with the CSIC of mus musculus brachyury, and we observed that there are shared genes enriched in myosin complex and migration, which actually is a way to infer that the ancestral function of brachyury was cell motility. We don't know what it's doing in Capsospora, but we know that in animals it's doing gastrulation, which is also kind of cell motility. So it's a complete gene regulatory network that is conserved from proteins to animals. Second example is the EPO pathway. The EPO pathway, which is now quite famous, but it was not known several years ago, is involved in organ growth. So it controls the growth of the organ. It's much more complex than this, but I just put three key genes: EPO, scaloped, and Yorkie. Scaloped and Yorkie, whenever they are phosphorylated, they are transcription factors, they activate growth. And the EPO is the upstream down regulator. So it stops, it down regulates scaloped and Yorkie. As I said, there are more genes, but let's focus on those three, because those three are present in the genome of Capsospora. Then we went to Drosophila. We went to the Iris Drosophila, one of the organisms in which EPO is best studied, best known. And we performed an overexpression of Drosophila scaloped, and Drosophila Yorkie. So the two transcription factors that, if they are phosphorylated and active, they continue to grow. And then what we observe is that we have an overgrowth of the eye. There is an overexpression of those, so we have an overgrowth of the eye. Interestingly, if we go to the Drosophila and we overexpress the Capsospora scaloped and the Capsospora Yorkie, it works very similarly. So we also have an overgrowth of the eye. And if we overexpress the Capsospora EPO, we have a shrinking of the eye. So it's actually down-regulated scaloped and Yorkie. So again, genes that are involved in a function, which is organ growth, and there are no organs in Capsospora, those genes are present in the genome of Capsospora, and if you put them in a bilateral organism, a complex metazoan, they seem to recover the function or do something similar. So what about gene regulation? So I remember what I was explaining that several years ago in some conference. I was explaining all these genes, how we sequence the genomes, all those genes, and how those genes involved in multicellularity functions were already present in the ancestor. I was explaining Brachyury and EPO and all of that, and I was very happy. But always at the end of the presentation, I will have always somebody that will, raise the hand and say, Iñaki, this is genes, but genes are just genes. So I think you are not looking at the important point. I say, okay, what is the important point? And he will say, the important point is you should check alternative splicing, especially if there is cell-type specific alternative splicing or exon shuffling. You should check that because maybe that's the key for animal origins. And then I will have, a woman just in front of me will raise the hand and say, Iñaki, this is very nice, but have you checked if there are transcription factor regulatory networks that are conserved? Because I think transcription factor regulatory networks have already been described as so important for animal development and for the formation of body plans. And then I will go to the coffee break. And when I was on the coffee break, I was going to take just a croissant, I remember. And then somebody will come to me and say, Iñaki, you should check animal-like histone code. Does it has epigenomic features? And then somebody will tell me, come and say, no, the important thing is not non-coding RNAs. MicroRNAs, Iñaki, microRNAs. I'm sure they don't have microRNAs. This is the key for animal regulation. This is the key for the complexity of animals, and so on and so on. And at the end, I say, okay, stop it. I want to come back to a conference. So I'm going to check all of that. So we decided to check all of that. And we check all of that specifically in Capsospora, but also in some of the other lineages. And we identified that Capsospora or Ichthyosporidium has all of those mechanisms except for enhancers. So those mechanisms were already present in the unicellular ancestor. And interestingly, those mechanisms are being used for Capsospora to transition from one life stage to another life stage. The way in which Capsospora transitions and decides to go to an aggregative stage or to a cystic stage is using the same mechanisms. So that's what we did. And the way we found that is because we went to those three life stages and we performed different analyses. So the first one is we performed a transcriptomic analysis, RNA-seq, of the different life stages. And we observed that they have different transcriptomic profiles, including alternative splicing, life stage-specific alternative splicing, cell type specific long non-coding RNAs. And then we went and performed proteome of all the different life stages, these cell stages. And again, they have different proteomic profiles with different specific functions. And then we performed the phosphopeptides for proteome. And we observed again that they have cell stages with specific phosphorylation profiles. So basically, including transcription factors that use different patterns of phosphorylation, depending if it's aggregates, cystic, or amoeboid. So basically, it has different phosphorylation sites, which are the ones that I'm marking here, like, for example, in P53, S492, with different grades of how phosphorylated they are, depending on how big is the ball and the color of the ball. For example, in P53, there is much more phosphorylation in the amoeba stage with the big green ball than in the aggregated stage, which is the small orange. So all of that means that the unicellular ancestor was quite complex, genetically complex. It had a complex regulatory genome, specifically for proximal regulation, and it had animal-like mechanisms of cell differentiation that are being used, or that was being used to transition probably between different life stages. And that's what Capsspora does. If you compare the transitions for every cell stage transition in Capsspora, it is regulated by histone modifications, by non-coding RNAs, by alternative splicing, and by a specific differential phosphorylation of transcription factors, which are the same mechanisms that animals use to differentiate cell types or tissues. Of course, to fully understand how the ancestral genes were recycled, and to understand, for example, the origin of genomic metazoan genomic regulation, we needed genetic tools. And that's what we have been doing in the last years, in collaboration with Elena Casacuberta, which eventually we have merged, and we are the same lab. So now we are co-PIs, both Elena Casacuberta and myself. We decided to work hard into that, and we have been able to develop transfection in 4 different lineages. And we have been able to develop CRISPR in coral autism. We are working more on that, but we believe this will be the first, and also the lab of Nicole King has developed transfection and CRISPR in guanoflagellates. So I think we now have the first functional platform to understand the origin of animals as never was possible before. So we can study things like the origin of animal development. We can study morphogenetic movements that they resemble some of the things that we see in animals, and see whether they share some of the molecular mechanisms with animals or not, whether they could be precursors of the morphogenetic processes that we see in animal development, including, for example, how choanocytes are. Choanocytes are found, so the multinucleases that we found in Ichthyosporeans are found in many other lineages, but cellularization from outside to inside seems to be exclusive to animals and Ichthyosporeans. Are they homologous or not? This is things that we can explain now, or we can try to address now with genetic tools. So basically, the first thing we did here, still with our genetic tools, is to check RNA-seq on the different moments. And actually, what we realize is that at the moment in which there is this polar layer of cells, like I told before, remember, this is a multinucleate. This moment in which there is polar layers of cells, there is an increase in expression levels of cell adhesion genes, such as integrins and actin binding proteins, which is the same that you will see in the Drosophila embryo at that specific stage. It doesn't mean it's homologous, but it's some kind of hint that could be potentially similar molecular mechanisms involved in those processes. We can also address the origin of animal cell types and the origin of spatial cell differentiation. And we are, for example, working now on that. And we want to know whether the colonies of guanoflagellates and the aggregates of Capsspora, which they look that they don't have cell types. They look like all the cells are similar, but we don't know if genetically are similar or not. So we are performing single-cell RNA-seq on guanoflagellate colonies and the aggregates of Capsspora to see whether spatial cell differentiation was already present before the origin of animals. We are also performing experimental evolution projects in which we are performing larger aggregates of Capsspora in order to check whether the larger aggregates of Capsspora will trigger more cell differentiation, more spatial cell differentiation, or additional features that we couldn't see before. And an important point is that we thought that we have resolved the tree of life in this part of the tree of life. So we thought that there are 4 unicellular lineages that are closely related to animals. Remember the 1, 2, 3, 4, Monoflagellates, Filasterian, Intersporians, and Colotician. But the thing is, we check environmental DNA and we observe that there are additional lineages on this part of the tree of life. That they don't look, at least at the molecular level, at the 18S sequence, they don't look like Monoflagellates or Filasterians or Intersporians or Coloticians or animals, but they seem to branch on this part of the tree of life. So one of the important things that we are going to do in the future is search for new unicellular lineages that are closely related to animals, or new animals that are earlier branching than sponges or ctenophores. Okay, so I have explained everything, all the data. The question is, remember the classical theory explained before. So do we have another theory? How did animals evolve then? What is our perspective on that? So that's where I'm going to go on. Based on everything that we have explained, I clearly believe that the Conoblastia theory, it has several flaws. First, we have observed that there was genetic innovation, but not so much. Second, we observed that at least the mechanisms of spatial cell differentiation were already present before. But there is another theory that has been quite forgotten by researchers, which is called the syntospore theory. This theory proposed that unicellular ancestor was not exactly like Monoflagellates, but was probably an organism with a complex and well-regulated life cycle. So it was transitioning between different life stages. And if you see the picture on the left, that's how they imagine the unicellular ancestor. And if you check the image, you will see that something resembles the Conoflagellates. They look like Conoflagellates and the colony of Conoflagellates. But then other things like the blue part, the aggregates, they resemble what I just saw on Capsospora. And then the other part above, it resembles quite a lot the Intersporians. So this is very interesting. And they propose is that there was a transition between temporal to spatial cell differentiation, just at the origin of animals. And actually, if you look at the organisms, we can see this thing. So we can see that there are unicellular relatives of animals, that they are flagellates, free-living, and they can form colonies, but we also see the amoeboids, and we also see the multinucleate cenocytes. So we see the flagellates, the amoeboids with phylopodia, and we see the multinucleates. So why not imagine then that there could be the possibility that there was a unicellular ancestor with a complex regulated life cycle? And what we see now are organisms that evolved from that ancestor, but they lost some of the life stages because maybe they didn't need it. So Conoflagellates maybe lost the amoebas and the multinucleates. The Intersporians lost the flagellates 1, and Capsospora lost the multinucleates and the flagellates 1. In fact, we have one organism now in the lab called Aveoforma whisleri that shows at least 10 different cell morphologies, and it transitions from one cell morphology to another extremely fast. It has kind of amoeba, it has kind of multinucleates, but in different shapes. It looks sometimes that there is a flagellate, so we have to look carefully. So why not imagine really that the unicellular ancestor was an organism able to do many different cell types? Depending on the environment, we change to one cell type or another. And it will have the mechanisms to transition from different cell types. If we have an analogy with the origin of the smartphone, that would be for me the Conoblastia theory. So you will have the landline telephone, which, I mean, most of you, or at least the ones I met yesterday, you haven't seen that, but I saw it when I was a children. you have a normal telephone landline, and then you will transition from that to a mobile phone that you could only go 2 meters away, and then some kind of advanced mobile phone. And then suddenly, the explosion of the smartphones. Suddenly, like suddenly the explosion of animals. If that was like that, it will mean, if that was linear, like I'm showing here, like the convolutional state theory, it will mean that at the origin of the smartphone, just when you got the smartphone, it will mean that you had an important technological evolution. It will mean that you had you got the GPS, you innovated for the first time camera, the photography industry, the internet connection, all of that. And you also had the capacity to regulate different technologies. But we know that's not true. We know that there was years and years of evolution of the photography industry. We know that there was years of GPS. The internet was already present before. Video cam industry, music industry. All of that was there before. So actually, in the case of the smartphone, what allows to have all of that is not that suddenly we innovated all these technologies. Is that suddenly we were able to incorporate all of them in one single entity, in one single machine that is able to develop all those technologies were already discovered in one single entity, which could be the one single multicellular. In the multicellular will be the same. We had already years and years of biological evolution, not industry evolution, but biological evolution of how cells can adapt to each other or adapt to other things, how cells can communicate, cell signaling, cell differentiation. You have different life stages. Genomic regulation, I've shown you, there was already genomic regulation. Different cell types, very clearly differentiated different cell types. All those technologies were there. The only thing is that maybe there was something new, maybe a software, biological software, that allowed to have different cell types and different cell behaviors at the same time, in the same organism, at different moments, maybe. And that's how multicellular animals appear. Is then the synthesis-spore theory the one we favor? The answer is no. Because the synthesis-spore theory is nice because it proposes, it's better in the fact that it doesn't propose only a conoflagellate like an ancestor, it proposes a unicellular organism with a complex life cycle. But the problem with the synthesis-spore is at the end, the transition from this this organism, it was again the conoflagellate because it has the homology with the sponges. So again, it comes back to the conoblastia theory, which I think is an important flaw. Okay, so if we have flaw in those theories, then how did animals evolve then? We see that there is a lot of technology before, but how was the transition specifically? What was the trigger? The thing is, we have a lot of problems here. There are several challenges to address this question. The first one is that the origin of animals occurred a long time ago, more than 607 million years ago. Very important, the origin of animals is a unique, non-repeatable, non-experimental historical event. And you know the problem with unique, non-repeatable, non-experimental historical events. You cannot experiment that on the lab. It only happened once. The origin of animals, all the data support this was only one single origin of animals, and never again. Then there is the problem, there is not a clear fossil record. Another problem, another challenge is evolution is not linear, which complicates backward reconstructions. Because things could go to complex or multicellular, and then lose multicellularity, and then acquire multicellularity again. And we don't see it because we don't have this information on the fossil record, for example, or in the molecular record. And more importantly than that, the most important of the challenge is the unknown. There is much more unknown than known. The analysis of DNA shows that there is potentially 8 novel lineages that are closely related to animals and that we don't know anything. Again, this is a formidable blind spot. We don't have any clue how they are. And if we don't have any clue about that, it's very, very difficult to address what was first, spatial cell differentiation, obligate multicellularity, embryonic development, What was first? We don't know, because now we have a big gap between unicellular organisms and animals. So we believe that under the current taxon sampling, it could improve, but under the current taxon sampling, we are observing a black swan event. And as you know, black swan events are so unique that it's almost impossible to decipher how they occur. You have a bias of knowledge, and you have a bias. This bias of knowledge tries to make you into assumptions that are not real, into reconstructions that are not real. It's impossible to reconstruct black swan events. And unless we get those taxa, we will continue to have a black swan event. And if we have a black swan event, the only thing we can be is be open. Some people think that it can only be through a conoflagellate that you get an animal. I don't believe so. I think there are several options to go from a unicellular ancestor to an animal. You could perfectly go from a conoflagellate, yes, an advanced conoblastea, a colony, but why not? You could go for a more multinucleate. Nobody has proposed that, ever, never. I believe that's possible. Once you have a multinucleate organism, you can pluralize that, and you can create an obligate multicellular organism. Nobody has ever proposed from aggregate. Actually, they say it's impossible to get complex multicellularity. I disagree. Once you have an advanced aggregate, you can have aggregates are very motile, they can move a lot, which is very important in order to create polarization, different structures inside. And once you have an advanced aggregate, then maybe you can go into clonal division. So I think we have to be open, because this is really a black swan event with all the unknown that we have. But we will try to get to those taxa. And I hope in some years, we will learn more. And if I learn more, I will explain this more to you and to everyone. Thank you very much. I finish here, but acknowledging the people from my lab, all the people that has done the work, the funding agencies, and all of you for listening. Thank you very much. And I'm glad to take any question that you may have.
Thank you, Iñaki. So for questions, please raise your hand and open your microphone. The people in YouTube, please write your questions in the chat and we will read them for you. Go ahead, Arno.
Thank you so much for this nice presentation. From plant scientist, I would say, I wonder if at the origin of animals, there is already, if you know, if there is already restriction of germinative cell lines, and if the life cycle is restricted to some haploidy level, and if meiosis is also asymmetric, if you have heterochiasmy that leads to anisogamy early on in this lineage.
Yeah, unfortunately, we don't know. We don't know if there is sex. We don't know if there is meiosis. We don't know if the meiosis, if there is meiosis, if it's asymmetric or not. It's something that we want to study more, and we hope we will get more information about that. For now, it's all a big unknown. But thanks for the question. Thanks for the question.
So we have a question by YouTube from Monica Garcia. She has really nice talk. Is it possible to infer what new functions Brachyury has in Xenopus based on the visible changes in embryo shape?
I'm not sure I get the question. So this is about Xenopus? If it's about Xenopus, I have no idea. If it's about Capsospora, then the function is cell motility, but we don't know specifically the function because we will need to do knockouts and other things. But if it's about the embryo Brachyury in Xenopus, I don't know. I did that in collaboration with an embryologist, and he is the one that will be able to answer this question. Unfortunately, I cannot. But thanks.
Okay. Go ahead, Enrique.
Oh, hi, Iñaki. Very nice talk. Thank you for all the information that you gave us. I'm really interested in knowing, is there any real unicellular organism? Because all the examples that you gave us, they are not completely unicellular. All cells, they have to interact with the environment, but also with other organisms. And all these models that you study are, they have at some stage, they are colonial. So in nature, does any organism can be considered really unicellular?
Well, in nature, thanks for the question. It's very, it's almost philosophical also, and I think we have to delve into that more. And from the plant side, from the animal side, from the fungi side, and so on. In nature, there are organisms that are considered unicellular. There have been not described a multicellular stage. One of the, I think, important points on those lineages is that all of them, they seem to have a multicellular stage. It could be that because early on they already incorporated new genes, it could be that they have more capacity than other eukaryotes to have these multicellular stages. And it could be as well, if you think about evolution, that there was several trials into multicellularity. Actually, multicellularity has been acquired several times, and maybe in the branch to metazoa, it was tried sometimes, but only one became obligate multicellular, which will be the animals. And yeah, we even hesitate should we call the unicellulars, because they do have always a multicellular life stage. But for now, we call it unicellular because they are not obligate multicellular.
Yeah.
But we always hesitate. So yeah, I agree, Enrique. I don't know.
No, because the interactions are quite complex for every organism. They cope with changes in the environment and all with changes in the interactions with other organisms. So that it's quite complex. Also, I have many questions, but one is very interesting that the Capsospora, they have Philopodia. Those are, and they look that they're very abundant. They are large too, because in animal cells we have very long Philopodia that are used for the interaction between cells. Have you seen that? Is there any communication activity among cells by using Philopodia or any other kind of cell extensions?
Yeah, we are not sure. We don't know, but preliminary data, visual microscopy analysis seems to suggest that the Philopodia are the ones, which actually makes kind of sense, are the ones that are being used to in the aggregation. So the Philopodia of one cell will go to the Philopodia on another cell. And actually, if you, for example, we have checked integrins by immunostaining, and integrins are localized specifically on several dots on the Philopodia. So they potentially are using those integrins on the Philopodia to attach to the substrate, and they could be using also for the aggregate. But we don't know about communication, unfortunately.
Yeah. Also, because maybe they can secrete some proteins that can be used also for communication.
No, I agree. And we check a little bit bioinformatically the secretome, and nothing was clear. So because it was not clear, I don't want to say anything. We don't know.
Yeah. Well, thank you very much. Very nice
Thanks
talk.
to you.
There is a question also by Luis Cardenas, and he asks whether the proteins required for multicellularity in animal cells are also required for plant cells.
Okay. No. So the functions are. So for example, this is very interesting because some of the functions are really needed for plants and for fungi. Transcriptional regulation, cell addition, signaling. But they seem, for some things, they seem to have evolved different genes. Of course, because they come from different proteins from different parts of the tree of life. It's very important, the raw material. So, for example, we checked several years ago the transcription factors, and transcription factors, they are enriched in plants compared to their ancestors or to the unicellular relative. They are enriched in fungi compared to their relative. They are enriched in animals. But there are different transcription factors are the ones that are enriched. So the transcription factors of animals are not the same as the transcription factors of plants. But the function of transcriptional regulation are needed in both of them and are enriched in both of them.
Go ahead, Andres.
Yeah. Thank you very much. My question is, has the capacity, no, has the I mean, this formation of colonies by these unicellular organisms under certain selective pressures can become heritable. Well, I was thinking that maybe
it can come heritable, and the way you can do it actually is by artificial selection. So if we do an experimental evolution and we try to force that they stay multicellular, or that they become larger multinucleates and so on and so on, you manage to do that. So there is a component of heritability in that. That's the way I see it. I don't know if I answered what you were asking, but that's how I see it.
Yes, thank you.
Christian?
Hello, it's a great talk, Iñaki. So I have a question with respect to the aggregation. So some years ago, there were some publications from Nicole King's group where they added bacterial membrane lipids and they caused aggregation of these guanoflagellates. And I wonder, I mean, in your models, you only talk about the, let's say, about the eukaryotic cells, different combinations that might have given rise to the animal or to the first precursor of the animals. But I was wondering, do you think these assays that there's some kind of artifacts, or do you think that bacteria or their molecules might have played a role in this, let's say, developing first animals?
Yeah, I think we have to check it. I think that's a potential possibility. At the end, the ecology is important. The ecology of the eukaryotes, they have prokaryotes around. For example, in the case of Capsospora, there is also a lipid and a calcium, and calcium. Both of them will trigger aggregation as well. So, and we have now a postdoc that is trying to understand whether interaction with prokaryotes will trigger aggregation and other things. So, we cannot take this out. So I think there is the potential that they could have played a role. However, I don't think we have to look for one specific bacteria. I think we have just to think that prokaryotes were there and they could have played a role in the fact of having prokaryotes. But it's not only prokaryotes. It's the environment, is the stress of the environment. Many of those things could have played a role. And I don't think so. Prokaryotes were before, and you didn't have the origin of animals. So it's a combination of things. But prokaryotes were there and they were important, but they were not the trigger because they were already before. So I think there was a combination of things.
Okay, thank you.
Yeah, thanks.
Any other question? Lot.
Hi. Hi. Can you hear me?
Yeah.
I was wondering, for these organisms, unicellular organisms that share many genes necessary for multicellularity, is there any chance that these unicellular organisms may have been multicellular in the past, but through evolution went back to unicellularity?
Yeah, I think this is potentially possible. Of course, they could not be obligated. Well, or maybe as well. I'm thinking maybe not obligated, but yes, as well. I think that's possible, and that's part of what I was saying. How difficult it is. It's part of the black swan event. We try to do things and reconstruct things in the way we think is logical, but sometimes things are not logical. And this is no black swan event. Whenever they happen, there is a lot of explanation, post-explanation. Like the crack of the market, in 1928, or in September 11. Then you reconstruct. Oh yes, there was something, but the fact is nobody had seen that before. Nobody had seen that before, and it's very difficult to reconstruct. And the same here. So it's potentially possible. For me, I'm open to that.
Okay, thank you.
I have a general question. So in Sphaenophora, the one that forms the silicium and then splits, if you were able to inhibit the step of when the cells become independent again, do you think you will be able to force that multicellular organisms to stay and live for a while, or
I don't know. I think that could be a potential, if there is a way to stop that. That could be a potential experimental evolution project. You try to fix it and then see whether then they become obligate. And once they become obligate, what did they do? Exactly. What did they do? Actually, we did an experimental evolution on Sphaeroforma, and what we were doing is trying to force them to be all the time bigger, sedimentation by sedimentation, all the time bigger. And eventually, we got 10 different lines that they once they explode, they stay together. So they didn't go and start, they stayed together. So it was always together, everything together. So it was kind of obligate multicellularity.
Okay. And my second question is regarding the self-recognition. Because at some point, probably mutations stop self-recognition and generate a new kind of variant of those multicellular organisms to form these big colonies. And so in terms of what you found in the genomics is basically integrin interactions, or there are reminiscence of proteins like the MHC in mammals?
There are MHC, I don't know what, but there are homologous of caderins, there are homologous of integrins, there are homologous of other additional ones, and there are some homologs, I don't remember what, that are like in monology, what cell recognition things. But cell recognition seems to be quite different in animals compared to our organisms. So, but yes, there are things of that kind. Yeah.
Okay. Gracias, Iñaki. There is probably the last question in the chat by David Gregorio. And his question is, could these proteins implicated in cellular aggregation be used as targets to disrupt cancer bodies? Is there any reference regarding the relationship between multicellularity and oncological processes?
Yeah, there are some, and actually we want to go deeper into that. I think it's very important. It resembles a lot. And so actually in cancer, whenever you have malignant cells, at the molecular level and at the behavior level, they resemble quite a lot organisms such as capsopora. And it's also an independent origin of kind of multicellularity. It's a reversion to multicellularity again. So I think there is a lot of potential cross-feeding between oncology and evolution, or at least evolution of animals. And we want to explore that further. So yeah, I totally agree with you. Yeah.
Okay, Iñaki, thank you very much. Muchas gracias por tu muy bonita presentación y además muy interesantes resultados. Claramente queda mucho por caminar y creo que a los chicos les abre un panorama nuevo que generalmente no volteamos a ver mucho en las áreas en las que estamos, pero muchas gracias por la presentación y gracias a todos por estar acá y por las preguntas tan interesantes que hicieron. Hasta luego, muchas gracias.
Gracias a vosotros, muchísimas gracias. Venga, buenos días, que vaya bien. Hasta luego.
Igualmente, chao.